And we can start.
Okay. Okay. Thank you. So now we are going to start the session. I think some of you might uh, be knowing that this webinar or this session that we are going to have today is more on that what are the new things that we can do using the computational techniques in especially in drug discovery. Okay, so my name is Avinash. So I'm the CEO of a company called Gloria Technology. This company is dealing in the computational biology research, and we are providing supports to experimentalists, to bioinformaticians, and to the theoretical biologists, so that they can conduct their experiment smoothly. Now, in this session, we have two more members from my team, Akshat and Suyesh. These are two members who are also going to give some kind of presentation in between. And in fact, Suyesh is going to start the first session. Now, before we start, I would also let you know that how this whole, 2 and 2.5 hour maybe will go, and what will be the plan to take it forward. So the design of this, webinar is in such a way that initially we would give some idea of, one of the very challenging but very important and critical technique in drug discovery, which is called molecular dynamic simulations. So of course we may not be able to do a hands-on that we regularly do on decode workshops, but today it is not like a typical workshop, so it is more like getting some information, and then you can definitely later on join our workshops on decode to understand or do more practice on those tools and techniques. But today what we are going to do is the first session will be to find out that what are the things that we can do on molecular dynamic simulations, and then we will take you through one example of protein simulations, and we will explain that how to do it. And in fact we can do it to some extent on our computer, but maybe you may not be able to replicate the same. If you can do that, that would be fantastic. But even if it is not, so the idea is to tell you that these are the steps that you can follow when you are doing this simulation, molecular dynamic simulation on your system. Then that will be the first session. In second session, I'm going to discuss about one of the tool that we have designed. Okay, so this workshop you may be knowing that even in the flyer or the information that we shared with you, that we developed a software or a platform which is called Analog. Okay, and that's why I think you guys might have heard this name when you were trying to join this workshop or webinar. So this Analog, the platform that we have designed, is definitely a in silico computational platform where you can perform several kind of steps that is required for drug designing and discovery. Okay, so as we understand that it might not be very convenient for the experimentalist, especially in biological area, to do some technical stuff on the computer. That also includes programming. Now, if but as we know that gradually and slowly everything is moving, or at least the bioinformatics is moving heavily towards machine learning, artificial intelligence, or complex computational technique that we need to know to perform our task. Now, what we can do when we do not have such kind of skill set or such kind of expertise, if we are coming from a experimental background, and we need to do some computational stuff. So what are the options in those cases? So one option is definitely to train yourself and then perform your task. But now training, it can only be up to the limited extent, because you cannot leave your core and then get yourself trained on something very new for you. So what we have done is we came up with a platform which is like very easy to use for anyone who is in this field, and we are going to explain that platform in some time. And then you can find it out that even applying the machine learning, molecular dynamic simulations, or any kind of mathematical operations, these things which sound very difficult or challenging in certain cases, could be very easy using the platform that we have developed, which is called Analog. So in second session, I'm going to talk about theoretical, that what are the attributes of Analog. And then in the third and the final session, we are going to see the demo version of it. Okay. Now, couple of things. Maybe initially, I would say, let you know, to all of you, that this is a commercial product. So this is a commercial product, means that if anyone wants this, they might have to go for the licensing, which might not be possible individually, but your institute, your department, your research lab, your project can afford it very easily, because this is like developed in a startup. So we understand that keeping the cost very high may not be a good business idea, of course, at the beginning. So this is the initial phase of Analog, and we will talk about, or so today we are going to talk about the technical aspect of it. Okay. Now, if one or many of you is uh, convinced with the technical aspect, or they think that okay, they want to use it for their research purpose, but before that they want a dedicated presentation of Analog in their department, in their um, college, in their research lab, or with the PI, or whatever, then we can arrange a meeting separately, and then we can go for it. Okay. So the overall idea here is definitely a soft launch of the platform, but along with it, it won't be like that completely marketing kind of webinar. It will be more on the science side, and then we are going to talk about that how the Analog platform can make those scientific processes very convenient and easy for anyone to use. Okay. Now, a couple of more things. So, before we start, so we just have had 10 minutes. So, I think we have enough time. Now, because right now, most of you are here, I'm not sure as the time progress, you may, feel like that it is now too much, and let's leave the meeting. I would request not to do that. So because we will keep you engaged, but we will try to keep you engaged, of course, but even then also we are not sure, that because we are not seeing you, so you are always free to shut down and go. But I will tell you that at the end of this whole webinar, and of course, I think the designated time is 3 hours, I believe, but it may not go 3 hours. Okay. And because I also understand that going for 3 hours, we can do it, but maybe the participants, you guys may not be able to sit continuously for 3 hours. So if we keep it continuous 3 hours, you may lose interest. And it is not like the things are not interesting or you are not paying attention. It is a very human kind of behavior, that after a certain time, you want a break or you want something else. So that's why let's keep it crisp, crunch, and also precise. And in this, even if we complete it in 2 hours or 2.5 hours, that is fine. So the whole idea is you should get some of the concept, and then also about analog, that what are the potentials of analog platform. And as I've seen, many of you, due to participant limit, yes, many of you may not be able to join because of the participant limit. Yes, Sujit, you're right. Um, but of course, the limitation is there. But I think Decode has also arranged a YouTube, I
Yes,
think,
sir. Yes,
live
sir.
stream.
We have shared on YouTube, and I have shared the link on the WhatsApp group also. So feedback form will be shared after the session in the WhatsApp group. So you can join by YouTube live streaming, and we will share the feedback form in the group. So there will be no issue in earning certificate. Over to you, sir.
Yeah. Okay. So there is a, YouTube thing already there, so you can just go and use that. If you are not able to, means some, I mean, if you are not able to join, so definitely you are not hearing me. But if anyone, your friend or anyone else, is not able to join, you let them know that there is a YouTube link, and you can go to the YouTube live stream. Now, what I was saying is, that I have seen the participants, and then of course the participants are from different regions, and different background, different qualification might be. So and some of them are also, I mean, not some of them, a lot of people. I don't know right now how many of you, but at least in the whole participant list, I have seen that many of you are not from India. So for Indian clients, so I'll come to this point when I will talk more about analog. Um, okay, so but the thing is, that for Indian clients, we are very much open towards the you know giving this platform, because this platform we are giving it with the hardware. Okay, so because some of the processes may not be feasible to run on a simple machine, so we have to design a customized hardware for it, and then we are providing it with the hardware. So for Indian people, right now it is feasible that we can supply the hardware along with the software. For outside India, we have not yet started. So for them, even for Indian also, we also have the service model. So in that case, if you think that you have certain things, and you want to just get it done, that also we can provide. Okay, so and then we can of course talk offline, that even if it is possible that if you really need that platform along with the hardware, we can arrange that one also. So we have a service model, and we also definitely licensing this product along with the hardware. Yeah. So yes, Fatima, do you want to say anything? Okay, it is just hello. Please allow the participants to record this session. Please, it's humble request. Okay, that I
So,
think.
sir, we will share the video recording. There is no issue. We have already streamed on YouTube, and we will share the video recording along with relevant document. No issue, sir.
Yeah, so all of you would get, but I think once you know that you will get it, the video recording, then you become, sometimes, not all of you, but sometimes some of you may become casual, because you are sure that okay, after the session I will anyhow going to get this. But okay, live is live, right? So recording is always a recording. You cannot ask anything, you cannot get engaged. Okay, So now I would allow Suyesh. So Suyesh is already here, and he is going to start the session with introduction towards the molecular dynamic simulation. Okay, and then I'll come back and talk a bit about the platform that we developed. Okay, okay, Suyesh, over to you now.
Yeah, thank you. So, hello everyone. Um, Hope everyone is doing great. So, um, I'll start with one of the most used and one of the most important tool in computational biology and bioinformatics, uh, which is basically molecular dynamic simulation. And we are going to see the overview of molecular dynamic simulation, as well as we'll have a hands-on session using the tool GrowMax. So I'll first start with giving a short presentation on the overview of MD simulation, like what it's actually is, and why do we use it, and how do we use it. So I'll just share my screen, and yeah, I guess my screen is must be visible now.
Yes.
Yes. So so what MD simulation or molecular dynamic simulation is. So basically, it's a technique in computational biology which is used to know more about the dynamics of of the molecule uh, of any system. It may be just the protein, or protein interacting with a peptide or small molecule. We can use any system, and we can define a condition for that system, and then we can know how how the system will behave in such condition. And that insight basically help us to know that how stable the system is. Like if we have a protein and a small molecule, then we can know if the interaction between protein and small molecule is stable or not, or if the protein which is there in the system is structurally stable or not. So so uh, so this is basically the intro of molecular dynamic simulation. And why do we do molecular dynamic simulation? Because we have to we have to know that if a particular small molecule will be effective uh, against a particular protein target or not, or if we have designed a peptide and we want to know that how this peptide is going to interact with our target protein. And even if we want to know that if the design peptide or design protein sequence is structurally stable or not. So there's a lot of application of MD simulation. And uh, and and this particular technique is being used in many sectors, like drug discovery, and drug repurposing, and peptide designing, and almost every in silico technique which requires a validation for structure stability or interaction. We perform MD simulation in that case. So now, what are the principles behind this technique, behind MD simulation? So it basically calculate forces based on normal Newton laws of motion, and uh, and along with forces, we also calculate potential energy. And based on these two calculation, we try to find out the coordinate of the whole system, uh, the coordinate of the whole system when it is in most stable conformation. And we use different equation for these calculation, and all these equations are provided through a mathematical kind of model, which is known as force field in this case. We also utilize some statistical tool or statistical analysis for different kind of calculations, and that is that is ultimately reflected in analysis for different simulation system. Now, how do we actually perform this MD simulation? So MD simulation basically has four major steps. So the first one is being solvation, in which first we define a particular area for our system. By the way, by system I mean the protein or protein ligand, anything which we are giving to the to the tool for performing MD simulation. So yeah, so first we need to define a particular region in which we are going to perform the simulation run, and uh, and define a particular uh, water water model for the system. We'll add the water model in the defined area, and check the charge on the system, and then utilize it. The solvation step itself consists of many minor steps, but the overall idea of solvation step is to add solvent molecules in the system, so that it will behave like or it will mimic the natural condition. Then the next step is minimization, in which the system which is being used for simulation is uh, is taken to the minimum energy state, which is also the most structurally stable state, and then that state is then used for further processing. In the next step, which is equilibration, the state of the environment of the simulation system is defined. So there are different ensembles. We can use different ensembles. Like here, we have given two examples like NVT and NPT. These basically defines the state of the system. We can set the system to be at a particular temperature or a particular pressure, and that temperature and pressure will be there for the complete simulation run, and that basically defines the state of the system. Then finally we have the production run, which is the actual MD simulation run, in which the system is it's yeah, the screen is still visible, right?
Yes, Suresh.
Yeah, yeah. So finally the production run is there, which is the actual MD simulation run, in which the system is allowed uh, to move freely in the defined in the predefined state, and basically at each step we take a kind of a snapshot which basically stores the coordinates or basically the confirmation of the system at that particular step. So we take snapshot at each step, and then we have a set of snapshot which contains the confirmation of our system at that particular step. So in a way we have a complete trajectory of the system, that how the system what was the condition of the system in the initial state, and how it transformed into a final and more stable state. So this is the flow of MD simulation, and and we'll see how do we perform each step, and what are the requirements of each step, and what are the outputs uh, of each step. So okay, there are some question in the chat box, so I'll just take them. So what is the purpose of doing simulation after dock? Okay, so docking docking also gives you if you are docking a complex of uh, protein and ligand, then docking will give you the most probable state in a most probable state in which the ligand is going to bind to the protein molecule. But further, MD simulation is performed to see what will be the most stable conformation of the complete complex in a fixed environment, in a fixed environmental condition. Then we have on what basis we are going to remove water molecule. Um, uh, we are not talking about docking here, but yeah, we need to remove every extra item which is there in the PDB file before performing docking. Force field is selected based on the simulation system which we have. Like if we have a protein, then we generally go for CHAM 36, which is more suitable for protein. But let's say if we have a heavy atom in our system, then CHAM is not defined for heavy atom. So we'll have to go for uh, amber or some other force field. Uh, yeah,
Suresh,
yeah.
just to intervene. I was just looking at some of the comments or some of the things that written in the chat box, and I assume that there are many people who are very new for molecular dynamic simulations, and in fact, to the in silico biology itself. So, as Suresh has already discussed about it, that what is the molecular dynamic simulation and all. Um, for conducting the molecular dynamic simulation, we are going to see that there is a tool called GROMACS. Okay, so GROMACS is the platform where we can perform the simulation. Then one of the question was, I think that was very initial question, that what is the in silico mean? So in silico means that the things that you perform on computer. So which is coming from the silicon graphics, which is used in the computer. So that's why it is called in silico. And one of the technique of the in silico biology is molecular dynamic simulation, where we understand, understand the dynamic behavior of molecule. Okay, so you guys are a lot of question about the docking. So because it's quickly relate your stuff with that you are docking something and then you are running the molecular dynamic simulation. But this is not just the complete application of MD simulations. There are many applications you may run molecular dynamics without any ligand, okay, which we are going to see today. Okay, and we are going to see on the GROMAC. So okay, so Suyesh, you can continue, and then if I feel that there might be some more question, I would just intervene and answer them.
Okay.
Please.
Yeah. This is the complete view of the flow in which we are going to perform the MD simulation in GROMAX. Then after completion of simulation, once we get the trajectory of the complete simulated system, we can perform different kind of uh, analysis on that trajectory. We can visualize. We can visualize the trajectory, like how the transition of the molecules has occurred. Then we can also analyze the structural stability of the complete system. We can see different state of the system, uh, of the complete trajectory. We can also calculate different energy terms, like free energy of the simulated system, or binding energy between protein and ligand. And yeah, we can do multiple analysis uh, on that trajectory obtained from the MD simulation run. And these analysis basically helps us to uh, know more about the simulated system and their interactions. So we'll see how to do MD simulation, and also some of the analysis. And we are going to use GROMAX for the simulation, which is the one of the most popular tool for protein and protein ligand simulation. So we'll see how do we perform different steps which I mentioned earlier in GROMAX. How do we select force field? How do we select different parameters which are required for the complete simulation run? So I'll move to the hands-on part now. And
I think you guys might be new to the GROMAX. Right now, Suyesh is going to perform the task, and parallelly, he will also explain those tasks. You may not be able to do it at the same time, so just go with him as we proceed, and then see that what are the steps. And then later on, if you want to try, you can try it. Yeah, this is a free application.
yeah, so I'll just share my screen, and I'll first show you what are the files which are required uh, before proceeding with the simulation run. So we'll definitely need a PDB file of protein uh, and ligand, if we have protein ligand system. I'm going to show it for a small peptide, so that we can finish it in time. Then we need a different MDP file. These are MDP file which holds parameter for different steps of MD simulation. So like em.mdp stores parameter for energy minimization steps, uh, ions for ionization, MD for the final MD run, production run, NPT and NVT for equilibration. So once I'll go through each step, I'll explain how these files are used in every case. Then we also have a force field file. This is not required unless you want to use a specific force field for your simulation done, because Bromax itself has some predefined force field, pre-integrated force field in the tool. But we can also use our own force field file. So I'll just start my Windows subsystem Linux. Bromax is only available for Linux. So to run Bromax in Windows, we'll have to have a Windows subsystem Linux, in which we can run Linux tools in Windows OS. So I'll quickly change my directory to this particular folder. Yeah. Once we are in this folder, we can see if all the files are there or not. Okay. Now we'll first start with having a topology file uh, for our system which we are going to simulate. So topology file basically contains the different data on the atoms present in the system, the bond, different parameter of bond like bond length, bond angle, uh, and and uh, and then and different molecules which are there in the complete system. So we'll start with generating our topology file. So and for that the command is gmx to initiate Chromex. Then the module I'm going to use here is PDB to GMX, and for input minus F or hyphen F is used for the input file. So here the input file is the PDB file of peptide, and we need to define an output file, and the output file will be in the format .grow. .grow because Bromax works with .grow files only. So I'll define an output file peptide.grow, and there are some other parameter also which we can define here. Like we can have our own name of the topology file, and that can be defined in -p, and let's say peptide dot top. But even if we don't use this particular flag -p, then also we can get our topology file name of topol dot top. There are also other parameters like hyphen water to remove water, or hyphen IGNH to to deal with the hydrogen present in the PDB file. So I'll use just this one, and when I'll enter, it will ask to choose a particular force field for the system. So we can go for the pre-integrated ones. like we have some amber, force field, charm, and bromos. Uh, but since we have charm 36 in our directory, we can go with charm 36. Then it will ask for the water model. Unless we don't have a requirement for a specific water model, we can go with the recommended one. So in this case, tip 3P is the recommended one. So we'll go with that. Now we have generated our uh, grow file and topol file. So we can see the change here. Now we have a topology file here and a grow file here. Uh, this ITP file is also like topology file, which includes topological parameter on atoms and uh, their bonds. Uh, and this ITP is called in the topology file, which will be used for uh, other other steps. So now we have the grow file which is required for GrowMax, and the topology file which contains the parameter uh, on the atoms present in our peptide. Now we can move um, with with the steps we just talked. Like the first step is solvation. So before solvation, we need to define a particular area in which we are going to add water and perform basically the simulation. So that part is done using another command which uses edit conf module of uh, GrowMax. And here in the input we give our uh, prepared grow file, and then for output we can define another uh, grow file. So I'll just name it pep and box, maybe, because we are going to define a box in this step. And then we'll include another flag here, -c or -c, which defines that the system or the peptide will be in the center of the box, and the distance of the box edge from the peptide is defined with -t, and that distance will be 1. And then we'll define the type of the box we are going to use. So we are going to use cubic box here, and yeah, and that's it. Now we have defined the box, and you can get the volume of the box. Here is the volume, and here other parameter for the box. Now once we are done with preparing the box for the simulation, then we can move forward with adding uh, solvent molecules in the box. And for that, I'm going to use another module of GrowMax, which is solvate. And here we'll define that our input file is pep box, and the structure file for water molecule we are going to use is SPC 216, which is already there with GrowMax. And we are using SPC 216 because this is a three-site water model, and the water model which we selected at the time of topology generation was also three-site water model, which was tip 3P. And now we'll define the output file here, and we can define it as maybe pip sol grow. Every output file will be grow only because GrowMax acts on grow file only. And now we'll also define which topology file should be used here. So yeah.
Suresh, just hold on 1 second. So I think you guys are writing questions on the message box, and it might not be possible to look at it right now when the session is going on. So what my request is that once one session will over, means let's say that we finish the MD, then we will keep like 5 minutes or 10 minutes, whatever, for the questions. Okay. So till that time, maybe you can keep your question with yourself, and because otherwise, your question will move up in the chat box, and we won't be able to see it, and we won't be able to address it, of course. So you just keep your question with you. I know that like I have seen one comment that it is going bit complex, and because it is complex, so you just stick with us, and you will get a easier solution by the end of the workshop or by the end of this webinar. So keep your question with you, and then once the one session is over, we will go to the question. Okay, Suresh, please continue.
Yeah, yeah. So now we have added the water model or water molecules in the simulation box, and which is reflected here. The number of solvent molecules is 4418. Uh, now once we have the water molecules, uh, we are going to neutralize the complete system. So for that, we are first going to uh, define the ions, and for that, I'm going to use another module of GrowMax, which is uh, Grompy, and Grompy basically takes uh, MDP file as input. So I'm going to give it the ions.mdp file, which was already there in the directory. And here I'll give it the uh, prepared grow file, which was prepared basically in the last step. So it was pep sol, and then again topology file here, and output file will be a TPR file. Yeah, and once this particular step is done, we'll further use another module, gen ion, uh, which takes the prepared TPR file as input, and the output will be another grow file. We can define its name as uh, maybe solve and ions dot grow, and again the topology file. Now we'll define what ion will use for neutralizing the complete system. So for positive ion, we are going to define it as P name, and we are going to use uh, sodium for positive ion. And for negative ion, we are going to define it by N name. And we are going to use chloride ion for the neutralization. And we'll define that the system needs to be neutral. And once it's done, the overall system is neutralized now. So with this step, the complete solvation part is done for the system. And now we'll move to the minimization part, where the system will attain a more uh, stable state, and the state having lesser energy. So again, we are going to use GMX Grompy module here, and in the similar pattern, the input file will be again .mdp, and uh, the latest prepared grow file. Now, again, the topology file with -p flag, and finally the output file in .tpr extension. So let's define it as em.tpr. Uh, sorry, it's gmx. Yeah, yeah. Once it's done, and the TPR file is generated, we are going to use that TPR file to perform our energy minimization run. So to performing this run, we are going to use another module of GrowMax, which is MD run module, uh, and will define uh, v just to visualize the run, and -def and m defines the name of the TPR file which was generated in the last step. Also, the output file will be named as em. So we'll just start the minimization run. It will take a few times since the system is not that big. It's just a peptide. So the minimization is complete. Uh, so now we are done with minimization. We can have a few plots here, like potential energy, to see how it's reduces, and and a couple of more plots on energy or other parameter. Uh, but but uh, but we'll move forward. We'll go to equilibration, and we are going to use NVT and NPT both equilibration. So I'll so let's see how do we perform NVT equilibration. So in a similar pattern, I'm going to use GMX Grompy module again, and again the input file will be MDP file, which is already there in the directory. We'll use the grow file which is generated in the last step, uh, and and also also the topology file again, and we'll define output file as nvt.tpr. Okay, we'll have to do some editing in the MDP file. So MDP file contains all the parameter required for the simulation run. Like it contains the the algorithm which we are going to use, the number of steps the run will take, and different constraints, and what's our reference temperature which we are setting for the system, and different parameters. You can go through each parameter one by one. For now, I'll just change this parameter. Since we just have the protein in our system, we can use uh, protein and non-protein here. We can also see the groups which are present in the system. Uh, for that, we'll just have to make a index file. So for making the index file, we are going to use make ndx module, and index file basically contains the component of the system which is given for the simulation. So we'll just prepare an index file, and it just takes input file here. We'll give em.gro, and so these are the system present in our simulation system, and since since the simulation needs to be performed for protein only, so we'll define protein, and rest other part like water and the iron uh, will be will be used in this group non-protein, and the same will be defined in the MDP file. So yeah, it need to be defined in the same manner uh, which is there in the index file, and just need to save it. We can always change the temperature or the total number of step for which the run needs to be performed. We can change these parameters also, but based on the system which we have. Yeah. So now once the MDP file is changed, we'll perform the run. Before that, I'll just save this index file, and to save it, we just need to type Q here and enter. We can also visualize the index file. Um, yeah, so this is the index file. It contains only the atom number of each group. So for system, all the atoms are included, and when we go down and like here non-water is there. So in this case, the complete protein, or in our case, the complete peptide is here- is uh, is defined here. So we have index for all the groups defined in different section of the index file. So yeah, now we have index file, and now we'll move forward with uh, NVT calibration. So again, the first step is to get the TPR file using the GROMP module. So we'll first run this, and again we'll use the prepared TPR file um, with the help of md-run module, and we'll define it as, we can also use -v here so that we can see how much time is left for the run to complete, and we'll define NVT here. So here it shows the total number of steps, and in picosecond and nanosecond how much uh, what's the total time of the run, and how the run is uh, being processed can be visualized here. So since the system is very small, just a small peptide is there, so the NVT run is fast. But for larger system like protein or protein peptide, protein ligand, it takes a lot of time. You can see the performance here. For peptide, it's 251 nanosecond per day, which means you can run the system for 251 nanosecond in 24 hours. But when when you will use a larger system like a complete protein or a complex of protein peptide or protein ligand, then this performance will go down to sometimes even lesser than 50 nanosecond per day. Yeah. So now NVT equilibration is done. So we'll move to uh, the second equilibration, which is NPT equilibration, to define the pressure of the system. And for it, again, we are going to use the same pattern. We are first going to get the TPR file using the Grompy module, and input again we are going to use the MDP file here. So npt.mdp, and here we are going to use the grow file which is obtained through the last step, and few more parameters like uh, like the trajectory obtained in the last step, and obviously the topology also. And now we'll define the TPR file here. Yeah, before that we need to edit our npt.mdp also, and we are going to use same set of group as we have used in NVT. So we can just copy it from NVT MDP file and use it in the NPT. So the pattern of the MDP file is same for all the run. And here also we have the groups defined here and other parameter. The only difference is in case of NVT, only temperature related parameters are there in the MDP file. But here in case of NPT, pressure related parameters are also there. But rest of the part is same. We have number of steps here, and the algorithm and other parameters. So yeah, once we have defined the group, we are going to use this command to generate the TPR file. And once we have generated a TPR file, we are going to again follow the same pattern and use the MD run module to run this NPT equilibration. So I'll just define NPT. Yeah. So these are the two equilibration steps, and with completion of this one, we are now left with just the production run. And production run also follows the same pattern. So we'll have to edit the uh, MDP file for production run too. So this is the MDP file for production run. So I'm going to use the same group here in this case. So protein and non-protein here, and this MDP file is also the same pattern. It has just a more number of steps are here. Rest the parameters are same. So maybe we can reduce it to maybe 1 nanosecond. By the way, this particular data is in a picosecond, so we'll have to do the calculation accordingly. Yeah. Now it's for 1 nanosecond. Okay, so I'll yeah the NPT equilibration is done. So I'll now perform the production run, and again we'll use the same pattern. We are going to generate the TPR file using the Grumpy module. In the input, I'm going to give it uh, md.mdp file, and the grow file generated in the last step. Also, other file like trajectory generated in the last step, and uh, defining the topology here, and the output as md.tpr. And once the TPI is generated, we are going to again use GMX MD run module to um, to perform the complete simulation. Yeah, so minus the and the name of our TPR file. Yeah. So yeah, it will take a few time, but by that time I can take a few question on this part, and then we'll move towards analysis part. So yeah, uh, it's getting complex. It is actually a bit complex. But we're going to provide you with a better solution for this one. But since this MD simulation is one of the important part of in silico study, so we can't just ignore it. Do we have to run NPD and VT together? It depends on your objective and depends on your system, which ensemble. There are more ensemble which defines the environment in different way. So we can go for that too. But since temperature and pressure are the one of the most important factors, so we use NVT and PT both. Yeah, these commands are standard, uh, and yeah, this set of commands basically completes a standard MD simulation run. When Bromex, yeah, okay, we can share that, but uh, if you will stick to the end, you will get a better alternative of that rather than going for these commands. You will have a better alternative. Yeah, we can always use another water molecule, another water model, and we can also use a different solvent other than water. You need to have a GPU if you want to perform the run faster. Uh, and yeah, that's and you need to have Linux also. No, both NVT and PT was performed, and yeah, let's see. See, okay, it's still running. So I'll take maybe a few more questions. I want to run the protein ligand complex simulation for 300 nanoseconds. Okay. For 300 nanoseconds, you can use any GPU. Uh, just the time taken for the run will be longer if the GPU is not that strong. MDB file in detail. It basically contains all the parameters which are required for for the simulation run, starting from the the number of nanosecond the run is going to be performed, the algorithm which it's going to follow, the constraints which are there on the system, and different parameters for temperature, pressure, what coupling method is being used. So everything related to that specific run is there in the corresponding MDP file. MDP file you can have it from the official GROMAX site, or you can also prepare it on your own if you know the uh, the command for each parameter. Yeah, Linux is necessary for this MD simulation because GROMAX uh, can be run only on Linux system. You can use Windows OS, like I'm using, but even in Windows you will have to have Windows subsystem Linux on which you will have to run GROMAX. Um, why was there a need to add ions? Yeah, we need to add ions to basically neutralize the whole system which we are going to use for our simulation run. What is the validation? Uh, for if you want to see that if the GROMAX is performing the MD in good way or not, we can always use uh, complex for which different parameters are already available. Different experimental studies are already available. So like we can have a complex of protein and ligand for which binding energy is already available, and we can validate that binding energy by calculating that after performing MD simulations in GROMAX. Is there no yeah, you'll have to use command prompt also, but yeah, you will have to use command prompt only, but you will definitely get a better alternative for it towards the end. Yeah. So now the run is complete. I'll just share the screen again. Yeah. Now the run is complete, and we can see what files are generated. So so these files are generated. The TPR was generated in the last step. Here CPD and XTC, which is the main trajectory file, the GROW file, which is the structure file in in GROMAX format, and then we have EDR file which contains details on energy related parameters. Then a log file which basically uh, has has stored each uh, the parameter and basically how each run has been performed. So we are going to use this particular XTC file, which is basically the complete trajectory file, and contains coordinates for each confirmation throughout the simulation. And we are going to use this for the analysis. So maybe I'll show you one or two analysis which are the most standard one. So first of all, uh, we need to clean our trajectory because the while simulation run, the system is free to move, so system might not always be present in the center. So we'll just clean our trajectory file, and for that we are going to use um, another module of GROMAX, which is GMX TRZ con, and we'll define our structure file as in minus s flag. So here we'll use the TPR file, because TPR file basically contains everything from structure, topology, and everything is stored in this TPR file. Then in input, we'll use our XTC file here, and in output, we'll define a clean XTC here, and we'll define different parameters for cleaning. First of all, we'll define that the system should be in center. Then we'll define how the system the all the molecules of the system will be. So and that is defined using this minus PVC flag. Yeah, we can use these two parameters, and we can run this. So it will ask which particular group needs to be centered. So we'll select protein here. That is the only molecule in our case, and it will ask for the output group. So we are going to take the complete system in the output, because we need to have other things also, like water and ants. Now the cleaning is complete. Now we'll see a few of the analysis which are which are the standard one, like the RMSD. So for RMSD, I'm going to use module of GROMAX, which is GMX RMS, which takes XTC as input. I'm going to use the cleaned one here, and again we'll have to give a structure. For that, I'm going to use the TPR file here, and we'll have to define output file. I'm going to name it as RMSD. The extension for the output file is .xvg, and yeah, we can. Oh, yeah. So it asks for the group for least square feet, which is basically the kind of the reference group which will be taken for the RMSD calculation. I'm going to take protein here, and now it's asking for group for the RMSD calculation. So I'll take maybe C alpha here. In case you have a protein and ligand and you want to see the deviation in ligand throughout the simulation, then you can take protein as the group for least square feet, and ligand as the group for RMSD calculation. So yeah, we have the we now have the RMSD file. It's here, rmsd.xvg. We can visualize it using different tools. I'm going to use XM Grace here. So yeah, so the RMSD will look like this. We have just 1 nanosecond run, so that's why it's appearing like this. But when you will have a run of 100 nanosecond or maybe 200 nanosecond, then you can get the kind of RMSD plot which you see in the manuscript. So RMSD basically tells how the system is deviating from its reference structure throughout the simulation. So on y-axis, this nm, this RMSD in nm represents the deviation at a particular time of simulation, which is defined in the x-axis. So lesser the deviation, more stable the system is considered, and and we generally take 0.3 as a standard cutoff for deviation in case of protein. So yeah, the system looks good. Yeah. So this was RMSD. We can also perform some other analysis, like we can perform RMSF. For RMSF, the pattern is same. Just the module will change. We are going to use RMSF module here. The input file will be same, the XTC file and the structure file. And for output file, we can define rmsf.xvg here. It will ask for the group for which RMSF need to be calculated. So we'll take just protein here, and we can also visualize this one in some grace. So RMSF basically tells how each residue present in our system is fluctuating throughout the simulation. So the y-axis in NM is representing fluctuation for each residue. The x-axis here is representing atom in this case, but we can also get it for residue. For that, we'll just have to add hyphen res here at the last of the RMSF command, and uh, now we'll get it for residue. So since it's just for a peptide, so the complete residue is maybe something 21, because it's starting from somewhere maybe 64 and ending at 85. So maybe 21 residue are there. So likewise, we can perform many different analysis. These two defines the structural stability of the system. There are other analysis for interaction stability, like binding energy analysis, and we also have free energy analysis. Some advanced analysis are also there, like PCA and all. So 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 considering the time, I'm going to stop here only. We have seen how to run complete simulation in GROMACS, and also how to perform uh, analysis uh, in GROMACS only. So yeah, this was the complete part for simulation in GROMACS. Maybe I can take a few questions, and then we can move forward with the other part of the workshop. Yeah, the time is set in the MDP file for the complete simulation run. So for each run, be it equilibration or production, and we can define the time for the run in the MDP file. Uh, we can use different techniques in MD, and we can also use some advanced techniques in MD simulation, like maybe QMMM for interaction. So yeah, there are many different techniques which can be used. This one is just the standard one. But more advanced, you will go more commands you'll have to execute. Can this process in laptop with 16 GB RAM? Yeah. If you have a GPU in your laptop, you can run these commands. Okay, I think that's it. We'll move forward with the workshop. So over to Dr. Avinash.
Yeah. Thank you, Suresh. So yes, so I definitely know that there would be many questions you guys need to know the answer of that, but definitely we have a limited time. We cannot go for each and every question. But overall, I have seen the questions in the message box, and it looks like there are many questions on the hardware. So Suresh already said that we need GPU at least to run this. It cannot, I mean, I'm not saying that it cannot run on the CPU, but then it's not a very good choice to run it on the CPU because CPU has a limited compute power, and the kind of the calculation that we do on molecular dynamic simulation may not be handled efficiently. It can be handled, but it may not be handled efficiently by your CPU compute power. Okay, so RAM is not that much important because this is not like a genomic kind of work. This is highly compute intensive. So here we need GPU. Okay, and GPU. Once we say GPU, there are certain GPUs which are coming with your laptop. You need to check it out whether it is listed in the Gromax site or not. Like Nvidia's GPU are definitely listed. Okay. Now, I know that there might be more question. You may ask the questions in the WhatsApp group or somewhere, like on the group itself, or you can just post your question on the YouTube. So people are posting the question on the YouTube also, and we are going to respond, once we see, or after this session, we maybe in a day or two we will respond each and every question. So what I would suggest is, those who are already joined here in the Zoom webinar, they can also go and join, or just go to the live streaming of YouTube and put your question at least. Okay. Now, let me go to the next part of this webinar, and which is like, that what is the alternative, and there is one alternative that we have designed, and I'm going to present that one, and then we will have a short demo of this, so that everyone there would understand that what exactly we are talking about. Okay, so this is the tool, or this is like the platform, that we have developed, and this platform is actually having two components. So the major components that was designed by us or developed by us, Gloria. And then there is a toxicity module also, which is designed by one of the UK-based company, Octane. Okay. Now, so this is a standard presentation. There might be some content which might not be very relevant for this, but I will just skip that very quickly if that is not relevant. Okay, and don't worry about the question. Once again I repeat that you just put your question in the live stream of the YouTube, because anyway we can look at it later on also, and we can respond it. So don't worry about the questions. Okay. Now, you know, so the company that we are here representing is called Gloria Technology. As you may know, that the whole platform is developed in Gloria. So this company is like founded in late 2019 in IT Delhi. IT Delhi, I think most of you, at least the people from India, definitely be knowing that the institute is, and even the people outside India may also know Indian Institute of Technology. So we have, in this company there are four verticals. So we are working in small molecule discovery. We work in protein design or biologics or peptide design. We also work in genomic data analysis, and then there are certain secondary data analysis that we perform. Okay, so now, this is like a very general slide, but with some updated numbers, and that might be interesting for many of you, and that also might be encouraging for many of you. So whenever there are challenges, then only the possibility of doing something innovative or doing something really great, right? If the things are not challenging, you won't have that room to do something great, right? So the drug discovery is a highly challenging task where you see that lots of people are involved in a R&D process, lots of time involved, capital involved, and even after all these things, the chances of failure is also very high. So it's like, a dead end from both sides, right? So it's like you are spending a lot, but then there is a high risk also that you have to take during the whole R&D discovery process, right? So one of the solution is using the computational tools and technique in a really appropriate way. Okay, and then that can reduce the cost, that can reduce the risk of failure, that can reduce the time investment, lot of things, and above all, that you can get the drug in the market in quick time, right? So that's the whole thing. I mean, we have seen during COVID that how critical the situation was, and people were really looking for some therapeutic or vaccine agent, and it was taking time because it is supposed to take time, but when this kind of discovery takes time, we have to pay life for this discovery, right? To discovery to happen, because in meantime you may lose a lot of life. So overall, we need to cut down the time, and that will cut down everything which is involved here. Okay, so if you see the proposed solution that we can come up with, so like computational drug design and optimization. So we talked about it in the presentation given by Suresh. Also, he talked about that how MD simulation could be useful there. AI and machine learning, which is like one of the very demanding and lots of buzz around, that how machine learning can help in drug design. Okay, and it's not 100% just the buzz, because now you can see, if you go and read some report, you will find that there are few drug compounds that are in clinical trial that has been discovered using the machine learning or AI technique or the computational technique. Data integration is also another thing which is very important, because if you want to speed up the whole process, so this is something where you guys can also get involved. If you are in early future, you can early career, so you can think about that, what you can do in data integration or application of machine learning in clinical trials. So uh, so lot of applications are proposed that can be used in clinical trial by a machine learning approach, and these are, I would say, relatively new. It is not like very new, but relatively new. If you compare it with the synthesis, then of course it's very new, right? So now now you also have right now 1 hour session on molecular dynamic simulation. There are many modules, right? Many things that we do in drug discovery using the computational technique or in silico technique. One of the technique is molecular dynamic simulation. Now, what you might have realized, the whole process, and I've seen some comments also, is bit complex, right? So it is, and I agree with you. I mean, it needs a good amount of training before you use molecular dynamic simulation efficiently. Okay, and this is just the one part. This is the simplest one, in fact, which we described. But the whole process is quite complex. You might also have seen it, or you even before today's session, you definitely have heard this thing before, that molecular dynamic simulation is really a crazy stuff. Now when this kind of things are complex and but at the same time required, right? So we cannot just skip it. because it is complex, we say that, okay, I'm not going to do this because it's complex. No, we have to do it because whether it is complex or easy, that doesn't matter. The thing which is important is that whether it is important or not. Okay. So if it is important, we have to do it. So as a company, what we thought is that because a lot of people are trying to get into this field, or a lot of people are already there in the field, and they are facing certain problems in the execution of these complex processes which is involved in silico drug discovery, right? So how about if we make those things easier for them? Okay. And in such a way that even the person who is from the experimental background can use it. And that's why we came up with this product, which is called Analog. Now, because some of the questions were on hardware, so as you can see in the picture, that this is not just a computational or a theoretical platform, but this is also coming up with a hardware, which is a workstation. So it's like a complete workstation you will get. Okay. And then you can perform all the tasks. So the thing is that you don't need anything extra or anything out of it. Okay. So whatever you need to perform, you can perform on this machine. And in fact, you can use the machine for some other purpose also. That is not somebody is going to stop you. But yes, whatever the programs you need, all programs would be installed. And then you would see a nice visual interface to interact with those programs, right? So what is the challenge? Challenge is challenge is like when you want to understand anything, for example, there are three ways, right? The first way is reading, right? When you read it, let's say that book. So when you read a book, that is the first way of understanding the thing. Second is then when you listen it, right? So when you listen it from someone, or you listen an audio or some kind of audio clip, that is the second mode of understanding the thing. And third is when you visualize it and also listening it, of course, so like a movie kind of thing. So you definitely would like to go for the third one, where you visualize it, right? So and if that is visualized properly and you listen at the time of the visualization, that would make a higher impact, right? So and you compare it with the reading. I mean, reading is a very tough kind of habit, or something which, not all of us can do it. So writing a command might be like reading kind of thing, right? While using such kind of tool, analog, would be like you are watching a movie, right? So and the same thing will happen, right? So whether you are reading or watching the same stuff on movie, your understanding would be very similar, right? But the overall experience would be highly different, right? Now in analog, it's like a complete package of eight different kind of models, right? So and we are going to talk about each module one by one. Okay, so just give me 1 second. Okay, so there are actually eight modules here, and as you can see, that this is like the welcome page or home page of this platform. So the moment you start this platform, you would see something like this, and you are going to see it in some time. And then there are eight different modules, and we are keep on improving and adding more and more. But right now there are these eight modules. What are these eight modules? We are going to see it. And definitely later on, we will give a live demo of the product, and then we will see that how it is easy, how much easy it is to use this kind of platform. Now, because many of you might be new, so I would also emphasize on the technical aspect or the scientific aspect, and then I would also say that what this tool is doing, right? Like the first module is QSAR. Now we need to understand, if we don't know what is QSAR, we need to understand what is QSAR. But one thing I'm sure that at least 90% of you definitely have heard about QSAR, which is quantitative structure activity relationship. So, what it is saying? It is saying that what is the quantitative way of relating the structure with the activity, right? And when we are saying quantitative, it means, because I'm used to take classes, so I take a lot of classes, so sometimes I may ask some question, but I would refrain myself in this webinar to ask any question, because otherwise we'll see a lot of responses on the chat box and I might not be able to see all of them. Then it is not fair to ask and not to see the answer. So QSAR is quantitative. So the first term is quantitative. So whenever you say quantitative, it means that mathematical, right? So, and when you say math, it means what? It means numbers, right? So, and why we want quantitative for anything, right? And the clear or the plain answer is so that mind can understand it in a best way. Numbers are easy to understand. If I'm running my car, and I'm driving my car, and I'm saying that right now the speed is very high, or right now my speed is low, that would not give you a good idea about the speed. But if I say my speed is 90 kilometer per hour, that will give you some idea, and then you can say that, okay, yes, the speed is really high, or something like that. So that's why whenever we present anything in number, it is easy to understand all the time, and that's how our whole education system based on, right? We give numbers, or we give scores, marks, to understand the potential of the candidate, right? And absolutely same thing we are doing here. We are giving scores, or giving some numbers to our candidate molecules, so that we can understand or we can relate its potential. And potential is what? Activity, right? And this is what the QSAR is. So in QSAR, what we are providing, and in fact in general also, that you need to go through a lot of machine learning way of establishing the relationship, right? So it is saying structure activity relationship and a mathematical model. So who will develop the mathematical model? Of course, we have to develop it, right? So if we have to develop it, it means that we need to go through all the coding part, right? It's only the, I mean, you cannot just develop it on page in pen and paper. So you have to develop it through code. And the moment all the coding comes, you would see that at least the 50% of the crowd will not be interested. And that's why here the QSAR, in QSAR, we are giving an interface to do the same thing using a simple click and filling some numbers in the graphical interface, and then develop a model. So it's like input. So you can see in the key features, it is saying input the smiles. Smiles are the representative representation of chemical entity, and then related activities. Suppose I know. So what is the question here? The question is that I know the activity of 1,000 compound, and I want to build a machine learning model so that in future it can predict the activity of any unknown compound. Okay, so that is the question. So that QSAR modeling can do for you. And here you just need to give the smile and the activity and go step by step, click, and very simple way to proceed. And finally, it will develop the QSAR model. That is the first module. Now, the second module is drug molecule generator. So this is really interesting. Here, what happens is that in drug discovery we screen a lot of compound against the protein, right? So when we are screening a lot of compound against the protein, so we need a lot of compound, right? So one way is to collect the compounds from any database. The other way is also to generate some new compound out of the existing compound. So this tool is, you might have heard this, that this image is AI generated, right? So like you definitely have seen it, that this image or the image of this person is AI generated. So actually that person does not exist. It is just generated. The image is completely generated by AI. Now here we are generating the molecule using AI. Okay, and that will resembles like a real molecule, and it will be a real molecule once you synthesize. Not in the human case, right? Of course, in human we cannot synthesize, but in molecule at least we can synthesize and we can get some new compound. So this is like generating new set of library out of the existing set of compounds. So that is what the drug molecule generator is. So I will not go like too much right now, because this is something like the introductory kind of webinar. And just to know that who else are more interested, we can take it forward and go in more detail. Then the third one is drug decoy identification. So what it does. So first we need to understand what is decoy. So drug, of course, drug is definitely the chemical entity that would have the therapeutic activity, right? That is called as drug. Now the decoy, what is decoy? So decoy is the chemical compound which looks like a drug, but actually it is not a drug molecule. Okay, so then how you differentiate? It is a very challenging task to differentiate between such compound and your drug compound. Okay, so we generated a machine, or we designed a machine learning model based on a method called graph neural network, and that can differentiate between drug and decoy. Once again, you don't need to do any programming anywhere. So all the programmings are inbuilt. You just need to put some parameters, and that also we will see that how easy it is to give the parameters. So you just give the parameter, say click a button, let's say it is called trained, and you click the button, it will be trained. Okay, and then you can use it whenever you want to, and that also you can use it through the analog platform itself. Okay, so it is like a drug and decoy. You want to differentiate a drug from the decoy. Now the fourth one is protein drug binding. What happens is, you guys know about docking, right? So docking is one of the very highly demanding, or highly known. I think it is one of the thing in the drug discovery which is highly known. Many of us know about docking at least. Okay, so what happens in docking is that your compound is going inside the protein at the active site or at the site of the binding. Now, when you perform the experiment, let's say that there is a solution of protein, and then you put your compound in that solution, then it's a very native kind of interaction, right? Like it is a very natural kind of interaction where the molecule would go and sit inside the binding pocket only if it is feasible, right? Now here it is not like anything real, right? There is no protein in reality on computer or the small molecule. So this is more like a geometrical fitting. Okay, of course there are many scoring functions involved with that, but more or less it is more like a geometrical fitting. So one of the limitation of docking is that it can almost dock anything with anything, right? Now you are not sure, like the people ask this question, that whether this docking is really a valid docking or not, or do we have any trust on the docking result. And of course, we have a very less or minimal trust, and that's why we go for simulation, right? But now here in this tool, we trained it on the experimental data, so that if you provide a docked complex to this tool, it will tell you that how native or how organic kind of binding it is, or or it is not. Okay, and based on the IC50, I don't know if you guys heard about this IC50. IC50 is nothing but a inhibition concentration for inhibiting the protein by 50% of its activity. So if I'm saying the IC50 of drug A is 2 nanomolar, it means that I need that drug 2 nanomolar to inhibit the activity of a targeted protein by 50%. And so if that IC50 is low, it means good, right? Because we want a drug in a lowest amount and highest activity, right? So we don't want to consume like 1 kg of paracetamol um, to regulate our fever, right? We just want a pill, right? So, and why it is doing it? Because in such a small concentration, paracetamol is active or effective, right? So we need the IC50 low, and that this tool will tell you that okay, the drug which is bound right now will give you this kind of IC50. Okay. The fourth one is, or I think fifth one is a protein drug interaction. Okay, so here what happens is that most of the times when we are talking about protein drug interaction, binding and all, we refer to a 3D structure. Okay, now 3D structure comes with its own complexity. What do you mean by the complexity? Is because, of course, I mean, if anything is 1D, it is simple. Once it goes to 2D, it is bit more complex, and when it goes to 3D, it is heavily complex, right? So what happens here is that if suppose I want to screen 1 million compound, okay, against my protein. Okay. Now, for screening, we know that we can perform docking. But what if the numbers are 1 million or 2 million? I don't know. So if such kind of large number is there, you might not be able to perform it using the docking. Okay. You need something which can handle the simpler form, and then then you can go for docking, of course. So it's like, I don't know, now it is not there, but few years back there was a screening program for clearing the IIT, and then there was a mains exam. So mains was like all the long answer question, and you have to write it down and everything. Now, why not the mains exam for everyone? Okay, why not it is for everyone? Because the number of the candidates applying for the exams are so high in number. So if you conduct a mains kind of exam for everyone, it will take a lot of time to evaluate the paper, right? Because you have to give the score, or you have to find out how the candidate performed. So that's why people have done, and is that they put the screening exam before, which is like the objective exam. Okay, and objective is very simple to evaluate, because you know that this is the correct answer, this is the wrong answer. And I think now computationally also you can check that answer sheet of objective paper. So this is like that, this is like you make but having said that, um, I keep on relating with the real example so that you can understand. But when you are doing the, let's say, objective exams, and suppose 10,000 people applied for that exam, uh, the complete exam, and then you put everyone initially for um, objective exam, and then out of the objective exams, those who performed best, you took, let's say, top 50, and said they are selected for the mains exam, right? Now, of course, it made the overall process easier, right? Now you can evaluate 50 candidates answer sheet, right, in mains exam. But there is a disadvantage also. What is the disadvantage is that you may miss the correct candidate because you applied one filter before, which is entirely different from the second filter. And once your pipeline is like this, so there is a chance. Of course, the chance is little. It's not too much, but there is a chance that you may miss a candidate for some or other reason. He or she might not perform very well in the objective exam, but he is the true candidate for the mains exam. So that can happen, but we have to live with certain kind of error, risk, all these things throughout, throughout the life. So now, same thing here. So because we want to screen 1 million compound, we cannot go for 3D, which is like a mains exam, or docking, which is like a mains exam, and simulation, which is like a super mains exam. So it's like interview kind of thing. Now, and that's why we have a simpler version where we take the smiles and we take the protein sequence and we screen them in a very quick way to get the answer. And this is what the protein drug interaction is doing for you. Now, the sixth one is antigen antibody, where once again it is just based on the sequence of antigen and sequence of antibody. This tool can give you that how strongly they are interacting or how weakly they are interacting. Okay. Now, the last two module, which might be very interesting for all of you, and I would ask Suyesh to give bit more emphasis on MD simulator and trajectory, maybe with some example. The way he did right now, using the GROMAX and the Linux terminal, you can actually do the same thing using this tool, MD simulator and trajectory analysis. This is based on the GROMAX itself, but but we have, made everything easier for the user so that he or she can easily perform the MD simulation in the MD simulator tool for protein, and for protein ligand. And then there is a trajectory analyzer which can analyze your trajectory and provide you like RMSD, RMSF, SASA, free energy landscape, and all kind of analysis it can perform for you using the simple click buttons. Okay, so that the trajectory analysis can do it for you. And maybe because you guys are right now fresh with MD simulation, you just have seen MD simulation. So I would suggest Suyesh to start the demo with the MD simulation itself. Okay, and then go reverse in the machine learning and AI based application. Okay. Okay, so I think that's it. And these are the case studies. It's not like that we just developed a tool. It's like that we developed a tool. We have given certain kind of services to other people, and they tested in the experiment, and they found that yes, the things that we are doing is actually working in the experiment. So it is not just a hypothetical stuff. Okay. There are one case study where we designed a small molecule. There is another case study where we designed the peptide, and both of them we have the experimental proof that yes, whatever we designed is working. This is our team. And Suyesh is already here. Akshat is also here in this call, and we all contributed for development of this tool. Okay, now I just stop here and would allow Suyesh, but before that, let me see that I can see a lot of questions. Okay, I think I just stuck on the Zoom. So you guys can hear my voice? I'm not. Suyesh, can you hear me? Okay.
So, dear participants, wait for 1 or 2 minutes. Dr. Nash will again live in 1 or 2 minutes. No, session is not over.
Hello. There might be some network glitch. So you can wait for 2 minutes. We'll just resolve it and get back.
So, we are getting back. Wait for 2 minutes and we will come back. Thank you. नहीं नहीं सर, वो मैंने तो आपको दे रखा है। अच्छा एक एक, एक सेकंड रुको सर।
So, guys, now we can resume the session. Yeah. So share the screen for the tool. I think all of you can see the tool now. And one more thing, because there are lots of questions. I think have been asked when I was presenting. Okay, so I just want to look at it. We can hear you. Okay. Sir, how GNN? Okay, how many GPUs? Okay. Okay, so is it possible that you can send all the questions? Um, how much cost to buy the complete setup? Is it subscription based? Yes, it is subscription based. We can talk about the costing. So, okay, so I'm just giving my one of the email ID, and you can drop, your query or anything regarding the tool. So, write the BD one, and also my own email ID. Hmm, Just first, right? This is the BD email ID, and this is my email ID. After you can drop your any concern or anything that you want to know about the product on these email IDs. And if you have any technical questions, once again, I would suggest that please go and type it on, or any type of question, please type it on the YouTube. And we will, because right now we may not be able to go to all the questions. It will take a lot of time. Okay. So we can just answer it there. Okay. Continue.
Okay, so now we'll move towards the demo of each module in the analog tool. So I'll first start with MD simulation. So I hope my voice is clear, right? Yeah. So so uh, so we have seen MD simulation using GROMAX, and we have seen that uh, first of all it requires uh, a Linux environment. Then you'll have to have a set of commands for it. But this particular module in analog, it doesn't require any command, and since the all the options are clearly visible, we can go through voices alone. Okay, okay, I'll do something about it. So okay, I guess now my voice
Yeah,
will
it's
be
okay. It's okay.
a bit more clear. Yeah. So yeah. So this is how the module for MD simulation looks. And as I also told during my presentation that there are several steps in MD simulation, and all the steps are included in this particular module, right up to the production run. And so I'll show you how it starts, and how you can move to different steps in this tool. So we'll start with uploading the PDB file here. So we just need to have a PDB file. No requirement of MDP file or force field file. we just need to have a PDB file. So we can maybe I can show you with one of the uh, sample file. And yeah, I have uploaded the PDB file. Now I just need to define which type of simulation I'm going to perform. So here it's just protein. So just clicking the option protein, I'll upload. The uploaded PDB file will be shown here. And now we'll move to other options, like if you want to remove water or non-protein molecules. So we don't have water in this uploaded molecule, so we'll just go for this part. Now the once the protein is uploaded and we have moved forward with these options. So now the solvation step is started. So it started with selection of force field. So we just need to select one force field. We need not have a separate file for the force field. We can just select the force field, and we can move forward. Here we can just select the water model and again move forward by just one click. It will itself perform the required analysis. Here we can give the distance, and for each parameter, a default value is being provided here. So you need not worry about what value you need to give for different options. So here the default value is 1.0. We'll go with that only. A box type, you can choose any. But by the way, for each option, you will have option to see what this particular section is about. So if you wanna know what the box, what do we mean by selecting a particular box type, so you can always hover on the question mark and see what's it's about. So we'll move forward. So every step in the solvation is performed in a single click. And now we are directly moved to the minimization part. Here in minimization also, we don't need to edit our MDP file. We can just select a few things here, like algorithm. We can use the default value of 5,000 here, again 1,000 here, and 0.02. If you want to go for a specific value, and if you know what can be input here for your system, then you can always use that. But you can always go with the default values. So now with just this single click, the minimization will be done. I have chosen the number of steps to be 5,000, so it might take a few time, but it won't take long. And similar setup uh, with GPU and installed required software will be provided with analog, so that will be each run is much faster than in conventional system. So now we are done with minimization. So you can see for a large system like this, the minimization is done in maybe 30 seconds or 1 minute at max. Now we have moved towards equilibration part. So we have options for equilibration in NVT, NPT, and production. And the options, since the MDP file which I've shown in each case looks same, just a few parameters are different. So likewise here we have similar option in all these three. So I'll maybe just show you this NVT part. And all the options have their default values. Also, the default parameters are already there. So we just need to give the input, and with just one click, the MVT will be done. So yeah, we can always roll back to the previous step, and uh, and we can perform that for our system. Monte Carlo simulation algorithm, like, it uses the classical MD simulation on GROMAX. You can also see the output which GROMAX shows you during the simulation in this particular window. So every time you will perform any step, like we have output for our minimizations here. So you will see all the output in this particular window. So I'll move forward. I'll show you another important module, which is trajectory analysis here. So while doing the trajectory analysis on the terminal, Linux terminal, I have shown you that XTC file, which is the trajectory file, TPR file, and topology files are required. So we are taking just those files in the input here. So you can upload your TPR file, topology file, XTC file, uh, and if you want to add index file also, then you can upload that too. And once the files are uploaded, you can define the system which is there. You can clean the trajectory with just a single run, and and there are a lot of analysis can be done here. So I've shown RMSD and RMSF during my presentation, but uh, you can also go for H bond, SASA, radius of gyration, binding energy, which will tell you binding affinity between uh, ligand and the target. Some advanced analysis are also there, like PCA and free energy landscape. And once you are done with all the analysis, you can request for report, and a report in this format with the plot you have used, and a detail about that plot will be generated, and you can get the report. Yeah. So every plot, right from RMSD, RMSF, H bond, everything will be included in the report. Even the PCA analysis part will be included, and free energy landscape and binding energy will be included in the report, and you will get a PDF file of it. Yeah. So coming back to the module. Not just analysis uh, on the uploaded system. You can also use your own XVG file. So sometimes what what happened, let's say we have a protein, and we have mutated the protein, and now we want to see how it behaves when it's interacted to a particular ligand. So we'll have to perform two different runs, one for control with wild type of the protein and ligand, and another with our mutated protein and ligand. So now we have a set of XVG file, but we wish to have those two XVG files plotted in a single plot. So for that part, we can use this particular section, all XVG plot, and we can plot it using this part. There's some question. This tool provide publication qualities. Yeah, the plots will be generated, will be publication quality only. Maybe I can show you a sample plot if it's there. Yeah. So the plot looks like this. The time is represented in picosecond, and RMST nm, and it looks like this. Also, if you will go for this option, all XVG plot, then you can get RMSD plot with multiple XVG at once. How are you going to obtain the topology file for ligand? You need not to worry about that part. That part is also that part is also covered in this tool. So you just need to uh, I'll I'll uh, I'll get back to the MD simulation module to explain that part. You just need to select this part, which is protein ligand, and you will have an option to upload protein file and ligand file, both structure file and nothing else. And the tool itself will calculate topology for ligand. How much time this tool generally takes considering. Uh, yeah, the time actually depends um, on the system, but for a standard kind of system, you will definitely get more than a performance of more than 100 nanosecond per day, or maybe 120, 130, maybe even more than that. Do we get you? Okay. So this was it for the MD simulation module and trajectory analysis module. Uh, in the trajectory analysis module, it's the same. You can hover to the question mark part to understand what this section is talking about, and you can select anything for your analysis. Like for RMSD, you can select protein and ligand here, or protein peptide. You can also define uh, what duration you want to calculate RMSD. Let's say if you wanna see RMSD for the last 30 nanosecond of the simulation, then you can uh, put the first frame uh, as the 70th nanosecond frame, and the last frame as the 100th one. And you can also define time interval uh, and unit of time in picosecond or nanosecond. Okay. Okay. This application. You need not to worry about system requirements to run MD simulation in this tool, because we are providing it along with the hardware, and the hardware is pretty good. It has the latest GPU, 30 GDX 3060 or 3090 will be there. So you need not worry about the performance of the tool. Can it deal with membrane also? That part is and soon you will get that option too in the tool. MD for peptide. Yeah, definitely. You can use any system with protein, peptide, protein, ligand, anything can be looking for umbrella simulation. Yeah, umbrella sampling is also in our pipeline. So in the next update, some advanced MD will also be added in the tool, which are basically steered umbrella sampling and MD for larger system like membrane. And can share the details. Yeah. You can just mail us on the provided mail ID, and you will get the detail about the cost and all. Okay. So I'll move towards the machine learning based modules in the tool, and these are quite helpful. Like let's say you want to design a uh, novel small molecule, novel drug molecule for a particular uh, particular target disease. Then
you can always collect the pre-approved drug for that. Let's say you want to design novel drug for Alzheimer. So you can collect pre-approved small molecules for that, and you can use this particular module of the tool. You can give the collected small molecules here, and you can define just a few parameters of machine learning model. You need not to worry about the terms. You can always get the detail on these terms, and we have also defined default values for each term in this part only. So you can give that those values uh, and generate a novel small molecule based on the sample molecules uploaded. What is umbrella simulation? Umbrella sampling simulation is basically enhanced sampling simulation technique. Yeah, yeah. So using this module, uh, you will get uh, generated molecules generated by machine learning model uh, based on the molecules which you have given. So and now let's say you have got a set of molecules with you, and you wanna know which one is basically active towards a specific target and which one is not. So for that query, we have another module. Yeah, voice is not here. I guess it should be a bit more clear now. And by the way, you can write all the queries in the provided email ID uh, about costing and subscription and all, and you will get the complete detail there. So yeah, I was telling that if let's say if you have a set of molecules and you want to know if this if the molecules in this set is active towards target or not. So this particular module will help you with this part. Uh, you will have to use a training data set here. You can give a training data set here, define the different parameter of machine learning here. But let's say you don't have a training data set, or you don't want to go with that hassle. So you can always go for the pre-trained model, and use the pre-trained model to directly classify your set of molecules as active or inactive, or decoy or drug candidates. So these two are very useful in case of small molecules. First one to generate novel small molecule, and second one to classify a set of small molecule as uh, decoy or inactive, decoy or active. Then let's say we have all the molecules. Uh, what is the file format for ligands to generate decoy? Generate decoy. You mean classify the molecules, right? So the format of the file need to be given here is mol2. So it takes basically structure file of the drug molecules as input, and based on that structure file, some features are extracted from that, and based on those features, it's being classified as drug or decoy. Yeah. So these are really helpful for uh, in case of small molecules. And let's say now we have screened out a fixed small molecule uh, for our use, or maybe we have 10 or 20 set of small molecules, and we want to know its binding affinity towards our target protein. So we can use this particular module of analog. In this one, we can get the binding affinity of a molecule towards its target based on the structure of uh, the small molecule, structure of protein, and we can also define a fixed pocket file in this case. So you can provide the ligand file in mol2 format, the protein file mol2 format, also the pocket file in mol2 format, and a target value for this is for training, and after training you can get the predicted value for your set of uh, query ligand files using this one. What would be the estimated cost? You can mail your query regarding the cost to the provided mail. Maybe I'll write the mail once again. Yeah, you can mail your query to this one. Uh, yeah, you can get the files from databases, but even if you don't have files, then we have a pre-trained model in most of our machine learning based modules. So yeah, this part was for structure based binding affinity prediction. Now let's say you don't have structure, you don't have a modeled target protein, then you can go for sequence based binding affinity prediction. You can give a sample data set for training, and let's say you don't have a data set uh, for the training purpose, you can go for pre-trained model and directly predict the binding affinity by giving your sequence of protein and the smiles of the small molecule. And you can also give the sequence here and smiles here if in case you just have one set of target protein and small molecule, and you will get a predicted score along with uh, you will also get accuracy for machine learning model in each of the module. So once you have done training, you will get you will get accuracy value as well as a plot to show the accuracy of the module. Okay, so we have got a novel drug here. We have predicted affinity for the drug using these modules. Now let's say we don't have a drug molecule, but we want to proceed towards uh, proceed with protein protein interaction or antigen antibody interaction. I mean, if you want to work upon uh, protein molecules and not small molecules, then you can use this particular module in which you will get binding affinity for your provided antigen antibody pair, and also if you want to test it for a protein protein complex, then that also you can do. Yeah, you will again you will have to give data for training, and if you don't have any particular data set for training, then you need not worry. You can just select a particular machine learning model from here and go for pre-trained model, and use that for your prediction. You can directly give the sequence of your antibody and antigen, and you will get interaction score or affinity score directly from the sequences. Uh, now let's say you have a small molecule, but you don't wanna predict binding affinity, but a particular property for the small molecule, let's say IC50, EC50, or something for the small molecule. Then you can go for QSAR modeling module, which we are already using it right now. It's open here, and you can upload your set of smiles here, uh, and and once the smiles are uploaded, you can decide which descriptor you wanna use for your analysis. You can also decide which particular model you want to use for your analysis. So there are some advanced models also, like random forest, Bayes model, decision tree. Uh, so you can just start with your smiles, and go with multiple analysis, like using different model and finding the best model, and using that for prediction, or you can use different descriptors also for the given set of smiles. So this particular module can be used for any kind of property for the small molecule, uh, something telling the interaction like EC50, something telling the effectiveness of the small molecule like EC50, IC50, or something which is telling the nature of the small molecule like solubility and all. So so uh, so yeah, this tool is a complete package for everything related to small molecule properties. So all in all, you can get the small molecules also. You can screen the small molecules based on their interaction towards the target protein. You can get the properties of the small molecules, and once you are done with uh, screening a particular small molecule, then you can go for its MD simulation, and then analysis, and finally getting uh, a complete report for your in silico study. So yeah, analog provide you a complete package for drug discovery research research work. So does analog also have pharmacophore? Is also in our pipeline, and that will be added pretty soon. Yeah, maybe before enhanced sampling, maybe before enhanced sampling also. Yeah. So I'll take maybe a few more questions, then we can wind it up. If you have any doubt regarding any particular module, you can ask. Um, in protein drug binding sensor shifting structure format, you can interact with generated. Yeah, you can provide all the all the mol2 file which you have, it may be of analogs of same small molecule, and that can be used to calculate the binding affinity towards your target. It's actually not just limited to QSR. It's basically QSPR. So you can use any property here in this particular tool. Actually, I'm performing. Yeah. Then definitely your system is more dense if it's taking 10 days for 100 nanosecond run. Yeah, then you can go for maybe better GPU, maybe something in range of 3000, GTX 3000, RTX 3000. Yeah. uh, where we can access this. This is not a website. If you wanna use it, use the complete analog. Uh, you can mail us on the provided email ID and have a complete detail on it. And drug decoy identification does generate ROC plot. It gives you a plot for accuracy, which is good enough to imply if the prepared model or the trained model is accurate or not. And you will get multiple values here, including R square, Pearson correlation, uh, also also mean square error. So you will get enough of data for uh, for the for determining its accuracy. So you need not worry about that. Okay, I guess that's it. You you can always contact on the provided email for any particular query.
So, Suyash, can you share your email? Some people are asking about analog. Uh, so they have queries regarding post and other details about analog. So can you provide a dedicated email so that our participant can contact you as per their requirement?
Yeah, I have provided it in the chat box. I'll
So
write
can
it
you
again.
once again type? I
Yeah.
will also share the in. I will also share it in WhatsApp group.
Sure. Uh, there's one more email ID which you can contact. Yeah. So uh, these are the email ID, and you can contact us through this for having more information on the analog and about its costing and subscription. Yeah. So that's it. Hope you have enjoyed the session, and please do contact us in case of any query. Thank you.
So kindly fill out the feedback form. We will deactivate the feedback form link after 8:30 p.m. Indian Standard Time. So you have around 45 minutes to fill out the feedback form. And we would like to thank the Grodia teams, Suyas, Dr. Avinash, Akshat for for delivering on our platform. Thank you. And for all analog related queries, you can always contact the Grodia team or you can contact us so that we can provide more details about this platform, which is going to be a booster in drug discovery. So thank you, Suyas. Now I seek your permission for ending today's session.
Yeah. Thank you, everyone.
Thank you. I have already shared the feedback form in the WhatsApp group.